Hi everyone, my name is Aaron Hoskins and I'm an associate professor here in the Department of Biochemistry at the University of Wisconsin-Madison. So I'm going to tell you a little bit about myself and my background and what my lab works on here at UW. So I grew up in rural Indiana, so out in the country surrounded by corn and soybean fields. I went to college and majored in chemistry at Purdue University. So after graduating from Purdue, I went to grad school at MIT out in Boston, where I again received a PhD in chemistry. And after that, I stayed in the Boston area and did a postdoc at Brandeis University and UMass Med School. So I was 4 years in undergrad, 6 years in grad school, and another 6 years as a postdoc. So after all of that, I moved to Madison and I started my own lab here in the Department of Biochemistry. And so these are some of my lab members at a baseball game a few years ago. So we are a group of undergraduates and graduate students and postdocs and staff scientists who are all interested in studying gene expression. And so, as many of you know, DNA genes are converted into RNA by transcription, and those RNAs are translated into proteins. And this is how the cell goes about carrying out its business. So in humans and in other eukaryotes, the RNA copies of DNA genes are not exact. And so the first product of DNA transcription is a precursor molecule called pre-mRNA. Then that needs to be converted into an RNA molecule that can be correctly translated into proteins. And so my laboratory studies this conversion of the pre-mRNA molecule into the mature messenger RNA for translation. So this process is carried out by a large nanoscale machine inside your body called the spliceosome that's made up of dozens of proteins and some RNA components. It has a molecular weight of 4 megadaltons, so that'd be 4 million grams per mole. And so it's a very large and complex enzyme that's essential for gene expression in humans. So we study splicing using a variety of methods. We do genetics and chemistry, and we also do physics. And so we do a lot of laser spectroscopy, and here is one of the laser setups in my laboratory that we're using to study RNA splicing. So one of the major questions we're tackling is how defects in RNA splicing can cause cancer. And so we're interested in this protein called SF3B1, which is frequently mutated in blood cancers like acute myeloid leukemia, as well as myelodysplastic syndromes. And mutations in this protein cause a defect in how blood stem cells turn into the different types of blood cells that make up your body, so platelets and white blood cells and red blood cells. And so we're interested in how mutations in this protein cause a defect in that process, as well as how you can target that protein with drugs for potential anti-cancer treatments. So with that, I'll wrap up, and I hope you enjoy learning about the University of Wisconsin-Madison. Bye.